Okay, so this is how to analyze ECG data in Kubios after you have filtered it in BioPack. So your first step is going to be to click on Open a New File, and then select the particular file you want to open and click Open. And it's going to prompt you to respond with what type of data set this is. So you'll enter ECG data as we're analyzing ECG data. And then from there, it'll ask you for the sampling rate of your ECG recording. So for example, this data was sampled at 250 Hertz. So I'm going to enter 250 and then press OK. So from there, you'll notice that there's a lot to look at within Kubios. So we're just going to go over a couple different things that you may want to pay attention to for your analysis. So down here in this bottom hand screen, you can flip through some instant results to look through. So for example, if you click time domain, it'll give you your mean RR, mean heart rate, RMSSD all automatically. You can also look at the frequency domain results and nonlinear results all automatically. So down here, this bottom hand screen is your results menu. So now up here, this is your actual data. So in this first chart right here, this is the R to R tachogram. And you can either zoom in or zoom out using the range function down here. So for example, if you wanted a closer look, you could decrease the range value. And if you want a more broad viewpoint of your tachogram, then you can increase the range value. You can also scroll through it using the scroll bar at the bottom to see more of the data set. So likewise up here, this is going to be your raw ECG waveform, and this is a little bit different, so you can increase the range, which is one way to zoom out and zoom in. You can also increase the axes over here. So for example, if you want to see the tops and bottoms of the waveform, you can increase your viewpoint over here. And then just like the tachogram, you can use the scroll feature to scroll through various portions of the waveform. So some other features that you may want to look at are in the settings over here. So if you click on the settings button in the toolbar, you can then scroll through the different presets and analysis options for time and frequency domain, nonlinear, and time varying, and change any presets that you need to change to suit your particular study. So for example, right now I have it set at 512 points for the spectrum, 150 samples for the window width, and 50% for the overlap for an FFT analysis, as these are the recommended 5-minute recording settings from Kubios. But for example, if you're looking at a longer length of recording, like a 24-hour length recording, you'll want to change these settings according to the methods of your study. Now another area you're going to want to pay attention to is the artifact correction method. So since we have filtered and corrected artifacts in BioPack, there shouldn't be any artifacts that need to be corrected, but even so, what we'll do is leave the setting on automatic artifact correction, and what this does is employs an algorithm used by Kubios to detect and correct artifacts. And so this is just an extra method of precaution that in case you missed something when you were editing your data and pre-processing it in BioPack, then the automatic correction method used by Kubios will then catch any missed artifacts that you were not able to correct and automatically correct them. So another key area to pay attention to is going to be the samples for analysis. So what this feature will do is allow you to select particular time frames from within your data to analyze. So for example, for this particular data set, there were 15 minutes total of data collected with 7 minutes of relaxed breathing and 7 minutes of paced breathing and a minute in between for adjustment. So for example, if we wanted to analyze the middle 5 minutes from each 7-minute section, what we would do is start here with sample 1, and you could label it. So for example, relaxed for relaxed breathing. You'll start the recording at 1 minute, and the length of the sample will be 5 minutes. And then you can click add to add your second sample. So for the second sample, we'll want it to start at 8 minutes and last for 5 minutes, and then you click OK. And you can also label the second sample. So now you can see in your tachogram, the blue highlighted region is the sample of data that's going to be analyzed. So we can see our relaxed sample and our paced sample clearly. So another area you're going to want to pay attention to is exporting your data. So when you're ready to export and you've selected your samples for analysis and have all the settings on what they need to be, then you'll go to File, and you have two options here. So if you're looking at just one data set, you could save your results as, and then you can either save them as a text file or a PDF. But if you're looking at multiple data sets from the same study, you can also append results to an SPSS-friendly batch file. And what this will do is, for example, if you're looking at 10 different data sets from the same study, it will save all of your analyses into one Excel spreadsheet, which will make statistical analysis much, much easier. So this is a really nice feature to use for when you're looking at multiple different files from the same study. But a key thing to note is that when you go to save it, you'll want to save it with the title of your study file, and then click Save when you're ready to do that. And in order to keep adding to it, you have to keep Kubios open. So instead of clicking the X when you're done with analysis, you'll go straight to File and then Open, and open your next data set after you've saved this one. Otherwise, when you close out and then re-enter, it won't save it to the same bash file, it'll save it as a completely new file. And that's the whole process for ECG analysis in Kubios.